So the continuation of this slide, we are going to discuss about the damage to the cell wall. The cell wall is mainly the principal stress bearing and shape maintaining element in bacteria. So it is the most important element in the bacteria for maintaining the shape, and its integrity is thus a critical importance for the viability of a particular cell. So for the viability and for the survival of a particular cell, the cell wall integrity is most important. In both gram-positive and gram-negative bacteria, this integrity of the cell wall, that means the scaffold of the cell wall, it is maintained by cross-linked polymer of peptidoglycan. In the cell wall of the gram-negative bacteria, this peptidoglycan layer is very thin, approximately 10 nanometers in thickness, and it is typically composed of 2 to 5 layers. But in gram-positive bacteria, the cell wall, that means the peptidoglycan layer, is much more thicker compared to gram-negative, that is 20 to 40 nanometers thick. This we have already discussed in the previous sections. While this peptidoglycan is the most important in providing the structural framework of the cell wall, teichoic acids, and structural framework of the cell wall, and the teichoic acids which are present in the cell wall, which make up roughly 50% of cell wall material and are thought to control overall surface charge. That I have already told, it provides the negative charge to the bacteria. So overall surface charge of the wall, and thus it may affect the murine hydrolase activity, resistance to antibacterial peptides, and adherence to the surface. So this teichoic acid is important in adherence to the surface, resistant to this antibiotics, and also to the murine hydrolase activity. Although both of these molecules are polymerized on the surface of the cytoplasmic membrane, and their precursors are assembled in the cytoplasm. So after the synthesis of this peptidoglycan, it moved towards the cytoplasmic membrane, that we have already discussed. Any event, even though these are things, any event which interfere with the assembly of peptidoglycan. Assembly of this peptidoglycan usually takes place in the cytoplasm. If any event occurs which may interfere in the peptidoglycan precursors and the transport of that object across the cell membrane, where it will integrate into cell wall, would compromise. It would result in compromising what? Integrity of the cell wall. If any of the events that interferes in the peptidoglycan synthesis, assembly, etc., it may affect what? Cell wall integrity. So the damage to the cell wall disturbs what? The state of cell electrolytes, which can activate death pathways. Approximately, it is also called apoptosis or programmed cell death. It may occur whenever there is a damage to the cell wall occurs. The regulated cell death and lysis in bacteria plays an important role in certain developmental processes, such as competence and biofilm development. They also play an important role in the elimination of damaged cells. What? Cell wall, such as those irreversibly injured by environmental or antibiotic stress. If any of this event which interfere in this peptidoglycan synthesis as well as assembly, it may affect the cell wall integrity, so the cell becomes dead. An example is an antibiotic, penicillin, which interfere with the bacterial cell wall synthesis. Penicillin is an important antibiotic which interfere in the bacterial cell wall. Thus, it may cause the cell, it disturbs the state of cell electrolytes, which activate death pathways within a cell. How the penicillin acts? It acts by binding to transpeptidases. So these are the enzymes which are present within the periplasmic space that we have already discussed, and this penicillin bind to these enzymes and inhibit the cross-linking of peptidoglycan subunits. Inhibit what? The cross-linking of peptidoglycan subunit. So as a result of the absence of this cross-linking, what happens? The bacterial cell in which the peptidoglycan assembly as well as synthesis doesn't take place. So the bacterial cell with a damaged cell wall occur, and thus the bacterial cell doesn't undergoes any binary fission, and the cell may result in the death of that cell. So by inhibiting the cross-linking of peptidoglycan, the bacterial cell with a damaged cell wall doesn't undergoes any binary fission, that is the multiplication of the bacterial cell doesn't take place and the bacteria will die. So these are all about the damage which are caused to the cell wall. So cell wall is most important in maintaining the integrity. Thank you.